Welcome to the Biomedical Podcast, the podcast that connects groundbreaking research with real-world answers. I'm Dr. Bensada, and in every episode, we examine new published articles to take a close look at the discoveries that are changing how we see science. We aim to fire up your interest and start a talk that goes beyond the lab.
Hey everyone, and welcome back to the Biomedical Podcast. We know it's been a little while since our last deep dive.
It has been a bit, yeah.
And we really appreciate you sticking with us. We're super excited to be back for our 6th episode.
Great to be back.
Today we're diving into the really mind-blowing world of organoids.
Fantastic topic.
We're going to explore how these amazing little lab-grown structures are changing how we understand and, potentially treat diseases like cancer.
Yeah, they're truly revolutionary. I mean, imagine growing miniature versions of our organs in the lab. That's basically what they are.
Mini organs, wow.
And what's really cool is that they can assemble themselves and even renew themselves.
Self-building.
Kind of, like self-building toys, but, made of cells. And scientists can grow them from different starting points, like basic stem cells or even cells taken directly from a patient's tumor. That self-organization was a really key early finding. Right. Now, for a long time, scientists studying diseases, they relied on growing cells in flat layers in a dish, those 2D cell lines.
The standard way, yeah.
But the big problem is, our bodies aren't flat. So these models, they miss out on the complex 3D structure and the whole environment surrounding cells in actual tissues.
Exactly, the microenvironment.
Plus, a tumor isn't just one type of cell. It's a whole mix, right? And those simple models often just don't capture that diversity.
True. And then you have animal models. They've been useful, incredibly useful, but there are basic biological differences between, say, a mouse and us.
Definitely. And ethical concerns, too, of course.
And that's where organoids are such a game changer. They really bridge that gap perfectly.
Okay.
They're much more like real human tissue than those flat cell cultures. And in many ways, maybe a better reflection of human biology than some animal models.
And you mentioned patient-derived ones, PDOs.
Yes. Patient-derived organoids, PDOs, these are particularly exciting. Think of them as personalized mini-organs grown from a specific patient's tissue.
Wow.
Which means they can actually mimic the unique characteristics of that person's disease, including all that variety of cells within their tumor. It's like getting a glimpse into your tumor's unique behavior, but in a lab dish.
Okay, so our mission then for this deep dive is to explore all the incredible ways organoids are being used in biomedicine. We want to focus on how they could really, transform personalized medicine, tailoring treatments just for you, and how they're speeding up the search for new drugs and, just helping us understand the basics of how diseases work.
So maybe let's start with a really important example, colorectal cancer, or CRC.
Okay, yeah.
It's still a major health challenge worldwide. Sadly, one of the top causes of cancer deaths, and surprisingly, the numbers seem to be rising in some groups.
Right. And what's super clever is how scientists are now making these CRC organoids. They can take a tiny sample from a patient's tumor, maybe during a biopsy or surgery.
And grow these 3D mini-tumors in the lab. And what's fascinating is that these little organoids, they keep the original tumor's characteristics, at the molecular level, the cellular level.
Yeah, they show really high homology, as we say. They're really similar to the patient's actual tumor tissue, almost like a little living copy.
A living copy, that's a great way to put it.
And this just opens up so many exciting possibilities for research, for treatment. One major application is creating better models to study the disease itself.
How it starts, how it grows.
Exactly. Researchers can use these CRC organelles to really dig down into how CRC develops and progresses. For instance, looking at how specific gene changes, mutations people might have heard of, like BRAF or P53, and others, how they affect the tumor's behavior.
Like making it resistant to drugs.
Yeah.
Or helping it spread.
Precisely. Things like survival signals for the cancer cells, drug resistance, metastasis. These organoids let us study those mechanisms much more directly in a human context. And it gets really interesting for finding new ways to actually fight the cancer, right? Beyond just the gene glitches.
Absolutely. These organoids are fantastic for basic biology research, too. Scientists can investigate the role of the tumor microenvironment, the TME.
That's all the surrounding stuff, the support cells and matrix.
Yeah, all the supporting cells, blood vessels, the scaffold, everything around the cancer cells. We can study how that TME influences tumor growth and spread. For example, how cancer cells interact with other cells like fibroblasts, and how certain signals like Wnt signals, think of them as communication lines between cells, play a role. Even tiny little bubbles or vesicles released by these fibroblasts can affect cancer cell growth.
Wow, okay.
And then there's research into how these mini-tumors get their energy, their metabolism, using organoids to understand how cancer cells gobble up nutrients like glutamine and what waste products they produce, especially those tricky cancer stem cells.
That directly leads to one of the most promising uses, I guess, figuring out which drugs will actually work for a specific patient.
Yes, and speeding up drug discovery overall. Because these PDOs are so much like a patient's actual tumor, we can test different drugs on them in the lab.
See which ones are most effective.
Exactly. Standard chemotherapy drugs, targeted therapies that go after specific molecular weaknesses, we can test them all. And some studies are showing a really encouraging link between how the organoids respond to drugs in the lab and how the patients actually did in the clinic.
So it's like a personalized drug trial in a dish.
That's a perfect analogy, yes.
So what does this all mean for, for you listening? Well, imagine a future where doctors could take a small sample of your tumor, grow these mini-tumors, test a whole bunch of different treatments on them, and then pick the one that's most likely to work best for you, potentially saving you from going through treatments that, well, wouldn't have helped anyway.
It holds huge promise for avoiding ineffective treatments and their side effects. What's more, organoids are proving incredibly valuable just in the process of discovering and developing new drugs.
Which is usually a long, expensive process, right?
Very long, very expensive, with a high failure rate, unfortunately. But organoids offer a much better, more relevant way to test new drug ideas early on.
See if they're effective before going into huge trials.
Right. And even finding new uses for existing drugs. That's called drug repositioning. Organoids can also be used to test if drugs are toxic or have unwanted side effects early on, giving crucial safety information.
Now, okay, we need to keep it real. Like any new technology, organoids aren't perfect yet, right? There are still challenges.
Oh, absolutely. It's important to be realistic. One big area people are working on is keeping the organoids stable in the lab over long periods.
Meaning?
While making sure they stay genetically like the original tumor and maintain that mix of different cell types, that heterogeneity. Sometimes cells in culture can change their characteristics over time. Maintaining that stability is a key focus.
Okay, that makes sense.
And another really exciting step forward, kind of tackling the simplicity issue, is developing what we call vascularized organoids.
Vascularized, meaning with blood vessels.
Exactly. These are organoids that actually grow their own tiny networks of blood vessels inside them.
How do they do that?
By including the cells that line blood vessels and endothelial cells in the culture. This gives the organoid better delivery of nutrients and oxygen, making them even more like real tissues.
Ah, I see. More realistic.
Much more realistic. And crucially, it allows researchers to study how tumors interact with blood vessels, which is vital for understanding how cancer grows and spreads.
Okay, so this is where we see how organoids are impacting way more than just colorectal cancer.
Definitely.
They're becoming powerful tools across the board, right? Other cancers, other diseases.
Yes, absolutely. Think about lung cancer. Scientists are now growing patient-derived lung cancer organoids, PDLCOs.
PDLCOs, okay.
To study how lung cancer starts, how it progresses, and, again, importantly, how it responds to different treatments.
And people are combining these with other new tech.
Oh, yeah. Really advanced technologies like artificial intelligence, analyze the complex data, 3D bioprinting.
Like printing the structure.
Sort of, yeah. Using bioinks with cells to create the structure. And also organoid-on-a-chip systems, miniature devices that mimic the body's environment even more closely.
Organoid on a chip, we should come back to that.
We should. And what's fascinating is how researchers are tackling some of those earlier challenges we mentioned, like the gel they often grow them in, matrigel. It can vary from batch to batch.
Ah, not ideal for consistent results.
Not ideal. So scientists are exploring more consistent, defined materials, called hydrogels, even some plant-based options, to create more reliable and reproducible ways to grow organoids. And there's a big push to standardize how we test drug sensitivity in these models.
Makes sense. You need everyone comparing apples to apples.
Exactly.
And what about other really tough cancers, like pancreatic cancer?
Yes, pancreatic cancer is another key area. It's notoriously difficult to treat. Organoids grown from pancreatic tumors are offering real new hope for finding personalized treatments and predicting drug response.
Are these being used with patients now?
There are clinical trials underway using pancreatic organoids to help guide treatment decisions. It's moving forward. And interestingly, The research is moving beyond just looking at, say, specific gene mutations to understand the overall state of the cancer cells, their transcriptional states. It's getting more sophisticated.
And I heard about adding immune cells, immune organoids.
Yes, that's a huge area of development. Creating immune organoids. These are organoids designed to include immune cells, our body's defenders.
Why is that important?
Well, it lets researchers study the complex interactions between cancer cells and the immune system right there in the tumor microenvironment. And crucially, test how well new immunotherapies, those treatments that boost the immune system to fight cancer, might actually work.
So you can see if an immunotherapy might work for that specific patient's tumor and immune cells.
That's the goal. There are different ways people are doing this, like combining different organoid types or using special cultural platforms. It gives a much more complete picture.
We're also seeing the development of organoid biobanks, like libraries.
Exactly. Like libraries or collections of well-characterized organoid models, representing different cancers, different subtypes, maybe different stages of disease.
And these are useful for?
Incredibly important for large-scale drug discovery efforts. If you want to test a new drug against many different types of lung cancer, for example, you can go to a biobank. And also for providing models for personalized medicine research.
Okay. And another powerful tool being combined with organoids is CRISPR, right? Gene editing.
Yes, CRISPR gene editing. This allows scientists to precisely make changes to the genes within the organoid cells.
To figure out what different genes do.
Exactly. To find out which genes are critical for cancer growth, or to understand how cancer cells become resistant to certain drugs. It's incredibly powerful for identifying new potential treatment targets.
You mentioned organoid on a chip earlier. Can we circle back to that?
Yeah, absolutely. Organoid on a chip technology. This involves putting the organoids into tiny microfluidic devices.
Microfluidic, like tiny plumbing.
Kind of. Tiny channels and chambers that allow precise control over the fluids and environment around the organoid. It can mimic blood flow, nutrient gradients, things you find in the body.
So it makes the model even more realistic than just a dish.
That's the idea. More controlled, more physiologically relevant for drug testing and disease modeling. It's a really exciting convergence of engineering and biology.
And is it getting easier to actually get the cells to start these organoids? I heard something about urine samples.
It is. For certain applications, yes. It's becoming possible to grow organoids from less invasive samples, like cells shed in urine.
Wow.
This is particularly relevant for cancers of the urinary system, bladder cancer, prostate cancer. It could make getting patient-derived material much easier and less invasive for research and potentially diagnostics.
That's amazing. So better ways to grow them, making them more complex, easier ways to get samples.
And improving the culture conditions generally.
Right.
Using those more defined materials we talked about instead of complex mixtures like matrigel. And also finding ways to make sure you're growing mostly the cancer cells, avoiding contamination by unwanted non-cancer cells from the original sample.
Right, you want a clean model.
Exactly. And finally, there's this field kind of bringing lots of data together called pharmacoproteogenomics.
Okay, that's a mouthful.
Pharmacoproteogenomics. Basically, scientists are looking at all the proteins, proteomics, and genes, genomics in the organoids, and linking that massive amount of data to how the organoids respond to drugs, pharmacology.
To predict drug responses better.
Precisely. To predict responses, understand resistance mechanisms, really get a deep molecular picture. There's interesting work on this in liver cancer organoids, for example, showing how this multi-omics approach can help personalize treatment.
So as we wrap up here, it's just crystal clear that organoid technology has this incredible transformative potential for biomedical research.
Absolutely. It really promises to revolutionize personalized medicine, no doubt about it.
Yeah.
And speed up the discovery of new and better drugs. Plus, giving us a much, much deeper understanding of complex diseases like cancer.
Yeah. And while, okay, there are still hurdles to overcome, making the models even more complex, standardizing everything.
Challenges remain for sure.
But the rapid progress we're seeing, especially, with adding in immune cells, blood vessels, combining with AI, CRISPR, microfluidics, it offers just a huge amount of hope for the future of patient care.
It really does. So maybe here's something for you, the listener, to think about. Imagine being able to study a tiny living version of your own organs, or perhaps a disease you might have, right there in the lab. How could that ability fundamentally change how we diagnose diseases, how we choose treatments, and maybe even how we think about health? It's a truly fascinating prospect, and the research, as we've discussed, is moving incredibly fast.
Definitely something to ponder.
Yeah.
Well, thanks so much for joining us for this deep dive into the amazing world of organoids. We'll be back soon with more exciting topics right here on the Biomedical Podcast.